Welcome to chapter 14, folks. This is going to be about cell movement and motility and contraction primarily. We'll be focusing a lot here on how motor proteins, which I'm sure you're familiar with, interact with the cell cytoskeleton. So let's get back a moment here and think about motility in general. If we're talking about cell motility, then that generally involves the movement of a cell or an organism through its environment, and it could also be the movement of the environment past or through a cell, like the beating of flagella on the surface of protozoa, for example. It could also be the way that components are moved within a cell. So cell motility has got many meanings, and its literal meaning has to be taken in the context of the system that we're looking at. Contractility, on the other hand, is used to describe the shortening of muscle cells, and that's really just a specialized form of motility. So we can then think about motility occurring at the tissue level, so for muscle contraction. At the cellular level, we could think about cell migration, and then subcellular levels if we're talking about organelles moving around within the cell. And so intercellular compartments can move as well. So, for example, microtubules of the mitotic spindle play an important role in the separation of chromosomes during mitosis in cell division. So to generate that movement, microtubules and microfilaments provide some kind of scaffold for motor proteins that do the work. They produce the motion at the molecular level. So we can really think about two eukaryotic motility systems before we start getting into some details. First one is microtubule-based motility systems, for example, the fast axonal transport you see in neurons, or the sliding of microtubules in cilia and flagella, and you'll be familiar with that at least to some level. And the second kind of motility system is going to be based on microfilament based systems, and so that would be, for example, muscle contraction. So we're not going to get into muscle contraction in too much detail, but we need to get into some detail so that if you're not taking physiology, you have some understanding of how muscles contract. So all this motility that we're talking about, Safa, is driven by a force generated by molecular motors. So those motors couple ATP hydrolysis to changes in shape and attachment of the motor protein to the cytoskeletal filament they're working with. And so those motor proteins undergo cycles of ATP hydrolysis, ADP release, loss of inorganic phosphate, and then acquisition of new ATP. And the consequent structural changes can be translated into molecular motion. When we look at molecular motors, we see common structural features, and we see that they invariably work by moving along a cytoskeletal filament, and they can go quite some significant distances. So let's think about some specifics. Let's start off looking at the role of microtubules and the motor proteins that work with those. So microtubules provide this, as you know, a rigid set of tracks for the transport of a variety of organelles and vesicles. When there is traffic towards the minus ends of microtubules, that's considered inbound. So remember, towards the minus end is towards the microtubule organizing center. That's inside the cell, close to the nucleus. Towards the plus end is considered to be outbound transport. So the plus end would be the tip of a cilia or the tip of a flagella, or it would be the periphery of a cell. So when we think about microtubule-associated motor proteins, we're talking about kinesins and dyneins. And so these proteins walk along the microtubules, and they provide the force needed for movement. So proteins that are produced in the cell body, for example, of an axon or of a neuron, are transported to the nerve ending in a process called fast axonal transport. And that involves the packaging of proteins into vesicles for transport. Then the vesicle is attached to a motor protein, the motor protein attached to a microtubule, and then the microtubule will walk towards the plus end of the microtubule. You can also see organelles being moved along microtubules through the axoplasm of a neuron. That's the cytoplasm of an axon, and that can go at rates of up to 2 micrometers per second. So kinesin 1, a protein called kinesin 1, which you see up here, That's involved in the ATP-dependent transport towards the plus ends, that's away from the centrosome, and that's often called anterograde axonal transport. Now, dynein, on the other hand, that's going to move cargo in the opposite direction, so towards the minus end, and that's called retrograde axonal transport. So, as what we're looking at on our slide here is three examples of some motor proteins. We're only going to worry about the kinesins and dyneins right now, and we'll come back to myosins when we look at proteins that work with actin filaments. The important thing here is that you can see that there are conserved structural features when you look at members of the motor protein families. So there's always this motor, which is shown here in this dark purple, and you can see it here in the cytoplasmic dynein, and then here in the heads of the myosin 5. There are often lever structures that you can see here and hidden back here. There's this stalk, which you see in kinesin 1 and in myosin 5. And there is some kind of tail structure. And then there are light chains when we look at this tail end of kinesin 1 and cytoplasmic dynein. So there are conserved structural features, so that speaks to some evolutionary conservation of the structural features of these proteins, and therefore some similar functions. Kinesins consist of 2 dimerized heavy chains and 2 light chains. So remember, those are proteins. So we've got 2 heavy chains which dimerize, and then 2 light chains. The heavy chains contain globular domains down here that attach to the microtubules. Then we have this coiled stalk region, and then a neck region. And then we have these light chains that make up the tail structure. And in the crystal structure of the protein, you can see that down here in the heads, the globular heads, there are binding sites for adenine-based nucleotides, and we'll talk more about those when we talk about nucleotide binding and ATP hydrolysis. So let's take a look at the movement here. Now, if you've taken general biology with me, you've seen me try and mimic how these things walk. So now we're just going to get into a bit more detail about the molecular mechanism of how these proteins walk, and the role of ATP, ADP, and inorganic phosphate in the movements. So kinesin movement looks like walking. It really does look like walking when you make molecular models of this behavior. And so the 2 globular head domains, those are the motor heads here, they take turns as the front foot. So you got to imagine them kind of doing this. And so they move along and they switch out whichever globular head is the front foot. The molecules are highly precessive, and that means they go through these repeated cycles of ATP binding, ATP hydrolysis, ADP release, and so on. And so they use a decent amount of ATP to walk relatively long distances before they detach. So if we think about the actual series of events of walking and break that down, what we have is the leading head. That's this one at the front here with this yellow kind of linker here. So that motor protein head there, it will bind ATP, and that induces a conformational change that causes the yellow linker region here to swing forward and dock into its associated head domain. So this swings forward and docks, and when it does that, it kind of flips the other head forward. And so the ATP binding here really causes a structural rearrangement of this head, which causes the trailing head to be flipped forward. So that binding of ATP there is flipping this foot forward. And so now the leading head here finds a new binding site, and that's usually about 16 nanometers down the microtubule, and it binds to the actin relatively weakly. So we've swung this trailing head forward, and that's been triggered by the binding of this ATP to the leading head. And so we've now got a leading head here, and the leading head, it's going to release a molecule of ADP, and that triggers tight binding to the to the microtubules. So I think I said actin just now, but we're obviously on microtubules. So that ADP release basically tightly links the foot or the globular head back down to the microtubule. So then the next step is what we have is the trailing head down here releases, hydrolyzes ATP to ADP, inorganic phosphate, and that inorganic phosphate is released, and that undocks that linker region that we talked about before, and then we can basically restart the cycle when we have a molecule of ATP binding to this now leading foot, which will cause this back foot to swing around, and then that foot will weakly associate with the microtubule. It will release its ADP, and then the trailing foot will hydrolyze its ATP, release phosphate, and then the front foot will bind ATP, and then we'll swing the back foot around. So it's just this processive repeating of this process, and so we can get the motor protein walking down the microtubule, and obviously it would be carrying something up here, some kind of cargo. So the things that get moved are diverse. The microtubule motors, dynein and kinesin, they're important for dynamically shaping and changing, for example, the endomembrane system. So extensions of the endoplasmic reticulum can be moved along microtubules. So here we have a kinesin moving this fragment of endoplasmic reticulum towards the plus end of the microtubule. We also see the vesicles to and from the Golgi. So down here, vesicles that are coming to and from the Golgi are carried by a variety of different microtubule motors, depending on which direction they need to go. If they are retrograde bound, then they would be on one type of motor, and if they are on anterograde directed transport, then they'll have a different type of motor. And so the cytoplasmic dyneins generally direct this retrograde transport, that's towards the microtubule organizing center and the minus ends of the microtubules, whereas the kinesins are going to deal with anterograde transport towards the plus end. Now there are some exceptions to this. For example, we met kinesin 13 a couple of videos ago, and that doesn't really have anything to do with the movement of material along microtubules. Rather, it acts to depolymerize microtubules. So most structures, most organelles in the cells have at least one motor protein associated with them, so that they can be moved if they need to be moved. But there are many different types of associations, and some of those associations are very cell type specific. So obviously we can't get into all of them. We're just talking about generalizations here. Okay, so we'll wrap it up there for kinesins and dyneins, and we'll come back in a moment to look at mechanisms of microtubule-based cell motility.